criminologists, welcome to our blog. I am Brendan Mark Supan, and today we will be talking about the scope and responsibilities of a forensic chemist. Without further ado, let's get started. First, let's define forensic chemistry. Chemistry, branch of science that deals with the study of the composition of matter and different changes that it undergoes. Forensic chemistry, specialized subfield of forensic science involving the application of techniques and principles of chemistry to the field of forensic investigation. Involves complex procedures of chemical analysis that are used to identify elements and compounds. Identification procedures are highly reliable. Identification is based on the physical and chemical properties of the substance, supported by the data obtained from analytical analysis. What exactly does a forensic chemist do? Analyze any non-biological materials found at a crime scene. Link the suspect to the crime. Determine the chemical makeup of the material, nature, and composition. Perform various types of tests depending on what the material is to find its origin. Prevent contamination of the sample during testing. Document his or her findings is an official record. Testify about the findings in a court of law. Now that we know the meaning of forensic chemistry, let's now move on to the scope and responsibilities of a chemist. After doing research, we summarize it and turn the information into 6 ideas. Hair samples are collected from the crime scene and suspects. The hair sample from the crime scene undergoes light microscopy next to the suspect sample. Depending on several factors, a technician can determine animal or human origin, growth phase, body area, racial group, sex, or age. Microscopy. Microscopy is the technical field of using microscopes to view objects and areas of objects that cannot be seen with the naked eye. Microscopy can be applied in the identification of trace evidence such as fragments, fibers, hairs, fingerprints which are left the crime scene on a victim or suspect. Typing. Blood samples from the crime scene are sent to the lab for analysis. Blood typing uses the principle of antisera interacting with antigens on the surface of red blood cells agglutination. Blood samples from the crime scene can be linked to the victim or suspect. If the samples are the same blood type, further molecular testing be performed. Blood typing. The use of blood in forensic analysis is a method for identifying individuals suspected of committing some kinds of crimes. When scientists identify blood types, they rely on slight differences in the antigens or proteins markers on the surfaces of red blood cells in a blood sample.
Toxicology. A urine sample is taken from a suspect and subjected to urine analysis. A typical urine test for most illegal drugs is done by immunoassay test. An antibody binds to a specific chemical causing for a color change. Immunoassay results can be confirmed using gas chromatography mass spectrometry. Toxicology. Forensic toxicologists perform scientific tests in bodily fluids and tissue samples to identify any drugs or chemicals present in the body as a part of a team investigating a crime. A forensic toxicologist will isolate and identify any substance in the body that may have contributed to the crime, such as alcohol. DNA fingerprinting. Biological evidence from the crime scene or victims is sent to the lab for analysis. Biological evidence can be blood, seminal or vaginal fluids, skin cells, etc. DNA is isolated from the samples using polymerase chain reaction and restriction enzymes. Fragments are produced that way are analyzed for matches among the samples. DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. sample from suspect. If the two DNA profiles are matched, then the evidence came from the suspect.
Santos Alberto from BS Crime 3118. Gunshot residue, scanning electron microscopy. An investigator uses residue collection kit of sent to swab a suspect's hand. A technician analyzes that sample using SEM energy dispersive spectrometry, evaluating then or gunshot residue. Gunshot residue analysis has revealed vital trace evidence consequence to criminal use of firearms. In forensic investigation, gunshot residue examination is used for solving a wide range of issues, from identification of a shooter to relating a bullet with the wound in reconstructing crime involving firearm. Chemical development of fingerprints. Evidence suspected to contain Latin prints is sent to the lab for analysis. The evidence can be suspected of different chemicals that reacts with the skin secretion, bringing out a print. Chemical development of fingerprints. Fingerprints and finger marks combine to provide the most powerful means of personal identification. Available to police and courts to recover prints can be manually compared with no antemoton records of research through an automated fingerprint system in order to verify or establish identity.
That's it for today's video. Thank you for listening. Hope you learned something and stay tuned for our next video.